Clinical Whole Solutions.
Thank you, Dr. Dangras, for giving us opportunity to introduce MGI, especially to most of the Indian customers. So, I'm Ravi Chilukoti from MGI. This is the content of our talk. So, as most of you are aware that MGI is a subsidiary of BGI. BGI is a big group of companies. It has several business units. MGI is one of the business units which has been established in the year of 2016. I will skip some of slides because I need to complete the talk within time. And this DNB-seq technology, it's called DNA nanobot sequencing technology. That's developed by a US company called Complete Genomics in the year of 2013. That has been acquired in the year of 2013. Then we optimized the technology to introduce our NGS platforms into the market. So DNB stands for DNA nanobot sequencing, and we use, for all our instruments, we use the DNA pattern arrays, and DNA nanobots will be prepared, then they will be loaded into the pattern arrays. Then we use 2-color and also 4-color imaging system. Especially for the G400 and T7, we use 4-color system. The technical advantage of our platforms is the accuracy of the data. 99.9% of the SNPs is possible, and also for the index we provide 99% accuracy and precision and also sensitivity. And the duplicate ratio, especially the competitors, they have the duplicate ratio more than 7 to 15%, whereas in our case the duplicate ratio is less than 3%. So for example, if you generate 1 TB data, mostly 30 GB is the maximum duplicate ratio, which will be unuseful for you at the time of analysis. And in our case, we use 10-base barcode, so that's why the index hoping is almost 0.0001. Coming to the sequencers, we have several units. Now, BGI-seq 500 and 50 are absolute. We have 2 series of systems, G series and T series. G series stands for the maximum throughput will be gigabase level. The G400, the maximum throughput is 1.4 TB, that means 1440 GB. G50, the maximum throughput is 150 GB. T7 is ultra throughput instrument. The maximum throughput is 6 terabase data within 24 hours. In our case, the 24 hours of sequencing time includes the demultiplexing time. Because to demultiplex 6 TB data, it generally takes more than one day time by using the conventional servers. But whereas in our case, the instrument control system software takes care of the demultiplexing also within 24 hours. And recently, we introduced benchtop systems, small systems, especially for the diagnostic labs. E series, the maximum throughput will be 20 GB. And for the automation of the library preparation systems, we provide 2 systems, SP100 and 960. For those people who have the sample throughput more than 20 per day, better to go for this SP100 because it can handle 16 samples for library preparation at a time. It can complete the library preparation within 2 hours, especially depending on the application. And for the people who doesn't have the bioinformatics software, we provide the Megaboard software, which is already having FPGA card, plus it's a hardware plus software. It accelerates the data analysis by 21 times. I will mention in the coming slides. For coming to the sample to report, we provide, for few of the applications, we provide total solution. That means we provide the kits for sample collection, library preparation, sequencing by using our instruments, and also data analysis, interpretation, and reporting. So that's why it's called total solution. We provide the total solution for NIPT, PGS, microbial detection in clinical samples, and also whole exome sequencing. That is also, we provide interpretation also using the Congenica software. As I mentioned earlier, we provide the sample collection kits, especially for the clinical samples, to collect the samples for stools. And we provide the nucleic acid extraction, especially for the microbial detection, DNA extraction, and also virus extraction, viral DNA extraction. And we provide the library preparation system, library preparation kits, especially for the whole genome sequencing, whole exome, RNA sequencing, epigenetics, single RNA, single cell RNA, and also epigenetic, whole genome bisulfite sequencing. As I mentioned earlier, the G50, the maximum throughput is 150 GB, it's old one. So the maximum read length at the moment is paired and 150. The minimum read length is single and 50. So it can complete the sequence string of paired and 100 within less than 48 hours. Whereas for those people who has major samples, especially for the whole exome or whole genome, we provide G400, which is ideal for the whole genome or whole exome transcriptome cancer panels. It has 2 slots. You can generate a data of 720 GB in one flow cell. So in full run, you can generate 1440 GB within 48 hours. The maximum throughput read length is paired and 200. As I mentioned, T7 it can generate 6 TB data within less than 24 hours. It has 4 slots. You can run either one flow cell at a time or 4 flow cells together. It will generate the 6 TB data including demultiplexing within 24 hours, which is really not possible by any other competitor at the moment. So at the moment we have installations in 36 countries. 34 TB database data has been generated within the last 3.5 years, because we entered into the market in 2017. So within 3 years, we were able to generate 34 database data. 740 publications are available using our DNB-seq chemistry, and 1400 plus installations are in 36 countries, and we have 460 plus users worldwide. I will skip some of the slides. And already we partnered with companies called Paragon Genomics, 10x Genomics to optimize their kits to be used on our platform. So coming to the clinical applications, this for those people who has the less sample throughput, DNB-seq G50 will be ideal, because the minimum throughput is 10 GB, maximum throughput is 150 GB. Minimum read length is single and 50, maximum is paired and 150. So we provide the flexibility to the customer to set up a run with less number of samples with 10 GB flow cell, 20 GB, 30 GB, and for those people who has the sample throughput and also who has the applications like whole exome, they can set up a run with 150 GB flow cell also. And coming to the quality, for the paired and 100, we guarantee 85% of the bases to have FRET score 30 or more. But whereas paired and 150, we guarantee 80% of the bases to have FRET score 30 or more. So at the moment we are providing the clinical total solutions for the NIPT, PGS, microbial detection, and also some cancer panels, and for whole exome sequencing also. So all our clinical total solutions are optimized using the GLIMS, because GLIMS is a laboratory information management system to automate the analysis also. So once the FASTQ files are generated after the sequence, the FASTQ files will be automatically uploaded to the Megaboard software by GLIMS. So that's why the total solution must have this GLIMS together with the Megaboard software, because all our reporting software are integrated with Megaboard software. And as I mentioned, for those people who would like to have automation for complete application, they can purchase SP100 or 960 also, because you can complete the library preparation within time and automatically it will be loaded into the flow cell. So coming to the chemistry, once the DNA is fragmented, you can use either Covaris, or for those people who doesn't want to use Covaris, we provide the enzymatic fragmentation also. The adapter plus barcode will be ligated to the fragmented DNA. In our case, there will be only one step of PCR, because adapter is already having the 10-base barcode. Because why we use 10-base barcode is for those people who pull 96 samples, the demultiplexing algorithm requires at least a difference of 2 bases between 2 samples. So if you use only 6 base or 8 base index for 96 samples, it is not possible to have 2 base difference between 2 samples. So that's why you provide 10 base barcode together with the adapter. After adapter ligation, only one step PCR will be there, then it will be single stranded. That then by using the splint oligo, the single stranded adapter ligated fragment will be circularized, then it will be copied to make 300 to 500 copies.
Ravi, you have 2 more minutes.
Okay, yeah. I will skip some of the slides. Yeah, by using this Megaboard software, you can accelerate the analysis by 21 times to 30 times depending on the application. For whole genome sequencing, 100 GB data, generally by using the GATK algorithm, it takes generally 42.8 hours, whereas by using Megabolt, we can complete the analysis and alignment within 2 hours. So as I mentioned, all the application reporting software are integrated into Megabolt. So this is one of the reporting, especially for the NIPT. It's called AccuR sequencing reporter, integrated into the Megabolt. I will skip this PGS also. Yeah, for NIPT, one step is important. Yeah. By using this G50, you can set up a run for NIPT of 32 samples. So once the library preparation is started, reporting will be completed with less than 24 hours. This is one of the report for one patient of NIPT. I will skip some of the slides. Yeah, for the metagenomics, we can identify the abundance of different microbes present in the clinical sample by using our total solution for microbial detection. So you can use whole genome metagenome or single bacteria also. For whole genome metagenome, if the sample contains virus, either it may be DNA or RNA virus. So we provide the DNA library preparation kit and also RNA library preparation kit. So it is also integrated with GLIMS. So once a saliva is collected, then it will be sample will be prepared and library will be prepared. Then once, okay, yeah. So once the sequencing is complete, automatically FASTQ files will be uploaded to the Megabolt software. The report will be generated and draft report will be stored in the system. Okay, this is one of the draft report. So our database for the microbial detection, it includes 20,000 microbe species containing bacterial, archaea, virus, fungus and parasites also. So that's why it reports the each microbe present in the sample and also abundance. This is our last slide. For the whole exome sequencing, we have two sets, 15 MB panel and also 16 MB panel. So this is called V4, 16 MB panel is called V5. So we provide the probes for RNA probes for the whole exome sequencing. So the advantage of our V5 panel is it covers the entire mitochondrial genome. So that's the advantage of V5 panel. So we partnered with third-party companies called SeqMax to generate the draft report after sequencing is completed. At the moment, SeqMax is available only in Chinese version. English version will be available by Q1 2020. So apart from that, we partnered with another company called Congenica. So the Congenica also will generate a draft report. Whoever purchases Congenica together with Megabolt, it generates a draft report for the whole exome sequencing of V4 or V5 panel. This is one of the sample.
You're using up your question answer time.
Yeah, that's fine. Thank you.